Okay, so today's Memory Decoding Journal Club is about a paper by Jinji Peng et al. In the Kevin Allen Lab in Heidelberg. And it's called Grid Cells Accurately Track Movement During Path Integration-Based Navigation Despite Switching Reference Frames. Just to give you a little bit of background, grid cells are functional cell types in the medial entorhinal cortex, MEC, that fire in a regular periodic fashion in a manner that's directly correlated with location and motion of an animal within an environment. They're usually either stellate interneurons or pyramidal neurons. And the Mosers, a husband and wife team, discovered grid cells in 2005 and received the Nobel Prize for it in 2014. In mouse experiments, valid stable grid cells are typically recorded when the mouse is moving at a speed of above 2.5 centimeters per second. That's important to know because these are motion-related functional effects. Unlike well-known place cells that fire in a specific location, place cells, you might remember them from the hippocampus stuff that we've done, grid cells tend to fire across positions in a triangle lattice. You can imagine them as a kind of coordinate system, and the distance within the lattice isn't the same or not exactly the same for each grid cell. And then you can imagine how if you have a bunch of different grid cells with lattices that are oriented slightly differently and shifted across space, maybe even at different sizes, that you can combine those. And with the maximum firing locations at those different shifted positions, you can define specific positions. So grid cells in MEC connect closely with hippocampal place cells to map and update spatial memory and cognitive representations. And that's led to models that propose how the activity of multiple grid cells combine to provide the receptive field input to place cells. Grid cell activity is essential for what's known as path integration, and the activity is observed whenever an animal is in motion. Path integration means updating a cognitive representation of location and orientation based on self-motion cues. And that's particularly useful for short periods of prediction when external landmark cues can't provide enough information. For example, you can imagine yourself taking some steps in a dark room to try to get to, I don't know, the bathroom or something. Traditional models of grid cell function assume that the grid cell activity is stable while an animal resides in the same global environmental context, so in the same, say, task box. And the authors in this paper recorded grid cell activity in vivo and demonstrated that grid cell reference frames switch even within a single behavioral trial, but continue to track movement. As I will describe below, grid cells re-anchor to task-relevant objects by translating their grid pattern, and a mouse's target can be predicted by a drift of the movement representation in grid cells. This is figure number 1. The first figure of the paper describes the 2D environment task. I don't know if you can see my cursor, but anyway, I'm trying to point at the first panel here, panel A.
We can see it.
Yeah, okay, great. And mouse performance on a so-called AutoPie task in dark and with lights on. AutoPie stands for Automated Path Integration. It's a protocol that was set up in other research papers, but they're using it here. As shown in the 4 boxes for the AutoPie task, those are the boxes down here, a mouse has to make its way, search, to a circular arena and search to locate a randomly placed lever. Then it has to press the lever. Then it has to find its way home to a base location, hence this is called homing, and there it receives a food reward. The 17 mice in these trials had been implanted with silicon probes recording from superficial layers of MEC, and those were either one H64LP NeuroNexus probe or between 1 and 2 H10 Cambridge Neurotech probes. I checked out the specs, or the spec docs for those probes, and there are a lot of different ways that the 64 recording sites can be situated on different shank packages for the probes, and it wasn't clear to me from the paper which arrangement they chose for that. The probe shanks were lowered to a depth of 0.6 to 0.8 millimeters in the brain. And then you can see here in figure A that when in the AutoPy task, the circular arena is connected with the home box through a small bridge. The sessions were set up so that the AutoPy trials were preceded and followed by random foraging in the arena. And during the AutoPy task, the mice were first given 7 trials in light and then alternating dark and light trials. The circular arena was rotated after each trial to one of 8 possible angles that are multiples of 45 degrees. And every 4th trial, the lever was moved to a new random location. And the combination of rotations and lever placements ensure that a new home base to lever relationship exists in most trials. In the final random foraging trial, there are 2 15-minute recordings that were made where the lever was placed in 2 different positions, and this was to account for a potential influence of lever on grid cell activity. They conducted between 3 and 25 sessions per mouse to a total of 180 and 38 to 172 recorded trials per session. The red lines show the path that a mouse is taking during a session. So you can see sometimes that's a very long path. Sometimes there's less in here. Figure B shows the path length of trials in dark and light conditions. And you can see that the path length is a bit longer in dark sessions, as you might imagine. Figure C shows the homing error in radians as a mouse tries to find the bridge to home on the arena periphery. As you can imagine, the errors are significantly greater in dark when mice are relying on path integration than in light. Still, in both cases, the errors were far below chance level. Figure D shows error as a function of path length. Error increases with the path length, and the error was independent of the lateral position of where the lever had been placed. Figure E shows the correlation between the search path length and the homing error, with a stronger correlation in dark than in light, as you might imagine. The position of the lever did influence homing direction, and more so in light than in dark. So from this, the authors conclude that mice primarily use path integration for homing in the dark, though there might still be some small influence of external cues. And before we go on, I wanted to ask if there are any questions about this task. Sounds like there aren't. Then we'll just continue. So the recordings were all made at 20 kilohertz sample rate. For quality, only spike clusters with a very distinct waveform, a mean firing rate above 0.1 hertz, and a small refractory period below 0.5, period ratio below 0.15 were analyzed. This refractory period ratio is the mean number of spikes in bins from 0 to 1.5 milliseconds divided by the maximum number of spikes in any bin between 5 milliseconds and 15 milliseconds. 5,746 neurons were recorded. 931 of those were classified as grid cells. Figure A shows examples of grid cell firing locations within the arena in each condition. So these are the conditions up here. In each condition of a session along with their firing rate. You can clearly see the spatial periodicity during random foraging. So it's very clear here. And surprisingly, this stable firing grid, well, I don't know if it's that surprising. We've seen this in other studies too. The stable firing grid was not observed during the AutoPy task. So it kind of drops away here. Figure B, down here, shows the spatial periodicity. So a lattice density for all of the grid cells at different rotation angles of the arena. And again, in different conditions. Figure C shows grid scores, and you can see that the same cells did not strongly exhibit the expected grid cell-like activity during AutoPy task. This was not because of the presence of the lever, as shown in random foraging with lever position 1 and 2, And figure D shows that the grid cells had a slightly lower firing rate during the AutoPy task. Figures E and F show lower peak firing rates and information scores in dark than light. The information score is calculated over the histogram of a cell's firing rates using the probability of residing in a rate bin and the mean firing rate. So in essence, it describes whether a distribution of rates is meaningful. If a cell always fires at the same rate, information is very low. If a cell fires at many rates about equally often, information is also low. Between those two extremes, you find firing rate distributions that are meaningful and that carry significant information. Now, the firing fields of grid cells appeared unstable during dark trials. The map similarity during AutoPy was significantly lower than across random foraging tasks, and that's shown in figure G. You can see here the difference is pretty pronounced. Interestingly, the similarity between two light trials was higher than between two dark trials. So if we compare light here, dark, or the two dark, the two light in this case. Sorry, that's what I actually meant to point to. Dark trials appear to lead to greater instability, but there is, even then, a degree of coherence during the AutoPy tasks, so the authors hypothesized that activity might still encode positional information. The authors found distinct apparent functions of grid cell firing during search versus during homing in darkness. And figure 3 here, so figure 3A, shows this for two grid cells. Those are the two grid cells. By plotting activity on the y-axis along with path y positions and lever y positions. So we have here the y positions, and all these gray bars indicate the path taken. And then we've got in black here the lever position. So this set of trials, these are all different trials, they've been organized according to where on the y-axis the lever position is located. And that way you can compare the lever position with these different trials, and also with the y location while running across a certain path. During the search, many grid cells fired at constant y positions. You can sort of see that here. They're firing constant locations on the y position. But then this changes a lot during homing. During homing, the firing field was predominantly determined by the distance of the mouse from the lever. And that did break down in light conditions, where some cells fired at constant y positions and others according to distance from lever. The correlation of firing rate with y position was higher during search, and the correlation with distance to lever was higher during homing, as shown in B, C, and D. So we've got that here. Again, we have, sorry if I can show that correctly. So the paths I indicated on the rows here, and you can see how this changes during homing. It's pretty clear. During search, so in figure E, let's see, that's over here, they compared correlations with distance traveled, y position, distance to lever, and time. So they brought in a few more possible correlations. And they found that during search, the correlation with y position was the highest, and during homing, the correlation with distance to lever was the highest, but the difference again disappears in light. So even though the grid patterns were not stable during AutoPy, the grid cells still provide some positional coding, although that can be influenced by the lever location. Next, the authors investigated stable similarities of temporal firing associations and firing phases between pairs of grid cells. The temporal firing association was defined as a Pearson correlation coefficient between the instantaneous firing rates of two cells. In figures 4A and B, so these here, we see this for two cell pairs, and we see a high correlation between cells 1 and 2, and you can kind of see that with your eyes as well, that those are pretty well correlated, and low correlation between cells 3 and 4. These associations were preserved for cell pairs between random foraging and autopie, shown in figure C here. So you can see that it gives you a nice 40-degree or something like that angle line. Figure D shows that these associations were also preserved across trial conditions. And figure E, it shows 5 grid cells. We've got them here along the axes. You've got, these are the random foraging maps of those grid cells along the 2 axes. And also here with the autopie, so we've got random foraging and autopie, the same thing. Again, across the axes are the 5 different cells. Then on the diagonals, you can see the autocorrelations. And then the remaining ones here, these are showing the spike-triggered cross-firing rate maps for pairs of grid cells. So this means that when one of the spikes fired, then you look in a very nearby window of time to find out when the other cell might have fired. That's how you develop these spike-triggered cross-firing rate maps. This assessed spatial offset of grid cells over short time intervals. Figure F shows that these are significantly correlated with spatial offsets that are largely maintained across conditions. We've got different conditions again here and comparing between the grid cells. And rotating spike-triggered cross-firing maps for one condition, shown here in G and H, shows that the correlation is most preserved at 0 degrees of rotation, and that implies that the orientation of grid cell firing maps was largely preserved across conditions. Because if that had changed when you test out rotating of one of the conditions, then you would have expected something to be a bit off-center here, not around 0. Together, this means that even though grid patterns appear absent during autopie, the temporal association and spatial offset of firing of grid cell pairs is maintained. So they're still kind of doing the same thing in terms of what their coding means and what another cell that has similar coding means. They're preserving this relationship between each other. So in terms of what we're going to get to momentarily, looking at it in a different manifold, a different spatial projection in a sense, in terms of the manifold of grid cell population dynamics, that's preserved here. Grid cell firing is known to be representable in that way as taking place on a toroidal manifold. It means that the collective activity of the neurons forms a continuous circular shape, like this torus here, as something that looks a bit like a donut in their internal state space. While individual grid cells fire in a periodic hexagonal pattern, so what you're seeing right up here, on the flat surface, the network of many cells functions as a continuous attractor network, where the population's activity patterns map smoothly onto a torus, regardless of the shape of the physical environment. There's a 2022 paper by the Moser Lab that describes this in detail. And you can carry out this mapping by projecting the 2D firing pattern onto a 3D space by carrying out a principal component analysis first. That's the PCA step here. And then reducing the dimensions because they retain 6 principal components, reducing the dimensions onto a uniform manifold 3D approximation using a UMAP. And the spikes for each individual cell are then located within a single patch of the new population state space. So there's one area on the torus where the grid cell will fire the most. To test directly if the grid cell network is performing path integration, even in the absence of a traditional grid firing patterns that we no longer see in that case, the authors built a computational framework to decode the mouse's movement directly from grid cell activity using this toroidal mapping. The goal is to see if one can reconstruct the mouse's path using only the neural data. Shown in A and B, so here and here, They needed to account for the geometry of grid cells, which function on a torus, as we just discussed, and they used the data from the first random foraging trial to define the grid's period and orientation. This allowed them to transform the mouse's Cartesian position, so the xy of the data, into a coordinate system within toroidal space. This is the V0 and V1 space. So there's a specific spot on that torus in V1 and V0 that is then the main location, the peak location for that cell. You can see the single location on the torus around which, if you see here, for example, the firing on that toroidal space, it looks like it's located in one particular place, and then the rate drops off around it. And then if you use the whole stack of cells that you recorded from, you can populate locations over the whole toroidal space. So shown in C, that's this part down here, it's got 2 bits. Within the toroidal coordinates, once you've established those, they trained a recurrent neural net to decode the animal's position based on the instantaneous firing rates of the grid cells. They then took the change in the decoded toroidal position over time and turned that into movement vectors. So vectors that with a direction and a length, and mapped those back into Cartesian space. And the final result is that they were then able to reconstruct the path that you see at the end here. So I don't want to rush on here. This is kind of important to understand. So I just want to check about thoughts, questions, anything related to this process. So everyone understands how we're mapping to a different space in 3D, and then we no longer have a hexagonal pattern. Instead, we have a single location, and then we can use the stack of neurons to describe anywhere on that torus, and that way you can then extract over time vectors that give you. Go ahead, Ken.
Yeah, I get this. I wanted to understand how head direction. So there's a head direction network that is parallel to this, that's keeping track of where the mouse is pointed in the overall space, I believe. And is there a good way of kind of understanding how that head direction maps onto this? It's like the head direction is going to push the bump in a certain direction at all times, right?
You'd think so. Yeah.
So like north is going to map to one of these, let's say the red axis of the donut.
Mm-hmm. Yeah.
And that's also going to be a direction within the head direction space.
Yeah, I suppose so. Yeah.
Okay.
I can't tell you exactly how the head direction network does that. The interesting thing about it is it would seem to say that you don't really need to go explicitly into toroidal space first, then create a recurrent neural network. You would be able to do it more directly in that head direction network, straight from grid cell activity.
Well, I mean, I feel that so the grid cell network here, this grid cell module, we're showing one module, right? So that's a bump attractor, and then there is a set of inhibitory neurons, excitatory neurons, God knows what, that pushes the bump a certain distance and a certain direction along the torus, depending upon the mouse's movements. And the way that the head direction cells are hooked up to that pushing network is kind of what I'm getting to. And it's I'm assuming at least that that's fixed. that there's a fixed relationship between the head direction network and the direction that the bump will be pushed. But maybe I'm getting ahead of myself, so I withdraw the question.
Okay, let's move on first. All right. Okay, to see how well this works, this is a continuation of the same figure. I've just split it up over 2 slides. In D and E, they compared the real path to the decoded path, and they measured the decoded directional error, so the angle between the real and decoded movement directions. And from this distribution, they extract 2 key metrics. They've got the directional precision. This is the mean vector length. It tells us how tightly the decoding matches the actual direction. And grid rotation, the circular mean that tells us if the grid representation shifted during the session plotted compared with random foraging. They first confirmed that the system is stable, as shown in figure 5F. There was no systematic rotation of the grid pattern between the first and second foraging trials. And this was for 49 sessions, recording sessions with at least 5 grid cells. They also looked at scaling. In 5G, you can see that the directional precision increases as more grid cells are added to the decoding. For sessions with at least 15 cells, the precision is quite high, comparable to sharply tuned head direction cells, and maybe that is somewhat related to your question, Ken. They then applied this analysis to the AutoPy task. And as you can see in 5H, here's the dark and light trials. The precision was significantly above chance compared with shuffled data distributions. Sorry, I'm actually getting ahead of myself. You can see this is actually just the reconstructed paths versus the real paths, and you can see that they're fairly accurate, not perfectly accurate reconstructions. And I just heard a bing, so maybe I should check that out first. Ariel, go ahead.
Yeah, apologies if this is a dumb question, but I think I follow the entire track of conversion from grid cells firing in XY space to mapping them on a toroidal manifold to eventually ending up with those positions for each cell in this toroidal space. I've either forgotten or never understood in the first place. How do you go back from the positions of all those cells in toroidal space to these locations back on XY space in the end here? Like, how are they generating these? This is what the grid cell says the position should be. Is it possible to briefly remind me of that?
I'm not going to be able to remind you of that precisely because I failed to look it up. I failed to look up how they actually do the calculation back into Cartesian space. I assumed it was just another mapping, just like they mapped to toroidal space, and that was possible. But now that I think of it, since actually they're still 2 by 2, so it's the same number of dimensions. It ought to be doable.
Yeah, I'm just struggling conceptually on the. Sorry.
This is the path integration problem, right? So how do they use the grid cell network to do path integration? In this case, in a computer as opposed to some other part of the brain.
Yeah. Do you want to go? Shall we go back to that? Take another peek at it.
Oh, they run it through an RNN. Okay.
So yeah, the RNN is the one that's interpreting, but this is still the RNN. If I'm not mistaken, they're not using 2 RNNs. So this is still the RNN that is taking that position, and then that is the one that's doing it. Yep.
Yeah. No, I do remember that now.
From here. Yep.
So they just feed the stack of cells in. They get into a black box, and they get the positions out once they've trained it.
Box. Okay.
That makes sense.
Fantastic.
All right. Thanks, Randall. Thanks, Ken.
Oh, no problem. Anyways, over here, I was getting ahead of myself a little bit. In H, we're really just looking at a comparison of the real path and the decoded path back let's see, that was 5H. So yeah, that is just that comparison. And it's fairly accurate, but not perfect, as you can quickly tell. When they compared the conditions, and this is 5I here, they found that directional precision dropped as the environment became more constrained, meaning as there was less information to work with, going from random foraging to the light trials of the AutoPy task, or as the task became more difficult, I guess, and finally to the dark trials. However, even in the dark trials, the precision was significantly above chance compared with shuffled data distributions, proving that the self-motion cues are sufficient for these modules to encode movement vectors. And in 5G, they confirmed the grid orientation remains stable across task shifts. And in 5K, they observed an interesting dynamic here. The error accumulation rate in the decoded path increased significantly once the mouse reached the lever, providing further insight into how path integration resets or interacts with goal locations. And that's something they wanted to study a bit further. So we saw in figure 5 that decoding accuracy takes a hit when the mouse reaches the lever. The authors hypothesized that this isn't just noise or error, but a fundamental change in how the grid cells are anchoring, moving from a room-based coordinate system to an object-centered one. So first, what they did is they looked at individual cell behavior near the lever. In panel A, you can see examples of grid cells that fire specifically when a mouse is at a certain distance and direction from the lever. We've got these direction and distance bits in here on the second row. And from these fields, so these fields appear consistently across trials, even though the lever location changes in the room. And to quantify this, panel B shows the directional selectivity of those neurons in a lever-centered reference. So in a lever-centered reference frame, that's significantly higher, you can see that in both cases, than chance during AutoPy. And this suggests that the grid cells are effectively treating the lever as a spatial anchor. In figure 6, they confirmed this by calculating the inter-trial stability of the firing maps. In dark trials, they found that the firing maps were much more stable when mapped in the lever-centered frame than in the standard room frame. And that's strong evidence that in the absence of light, the lever becomes the primary spatial reference. Now, in the next half of this figure, they return to the RNN decoding model to see what this looks like in the abstract toroidal space for the whole population of grid cells, or at least the ones they recorded from. In panel D, this shows the animal's path around the lever. And if you look at the decoded positions in toroidal space, let's go. Actually, we're kind of jumping straight down to the bottom here. You see that it forms a stable cluster. The grid module effectively locks onto the lever position, and they're showing that in that bottom row here as a kernel density estimate of lever position in toroidal space. Panel E confirms that this is not due to drift. The distribution of the decoded directional error stays centered around 0, indicating that the grid representation is maintaining a consistent baseline even as the reference frame shifts. And then a key question is, how does this re-anchoring happen? Does the grid pattern rotate or does it shift? And in panel F, this compares the orientation drift at the lever versus distant matched paths elsewhere. There's no significant difference, which tells us that the re-anchoring is occurring via translation of the grid pattern, not a rotation of the entire grid axis. Finally, they tracked the anchoring strength over time. And this is in panel G, which I'm sorry that I didn't blow all these up to individual slides. So at the start, you can see in that panel that at the start of the search path, the modules are firmly anchored to the room. And you can kind of see this progress here, right? 1, 2, 3. They're anchored at first to the room, and then that, the dominant anchoring shifts to the lever itself, and that decreases then again as the mouse departs. So this provides a clear time-resolved picture of the brain dynamically updating its navigational reference frame based on task-relevant objects. So we've established that mice use grid cells to path integrate in the dark and can re-anchor to a lever. But path integration is an iterative process that relies on summing up movement vectors. In the absence of visual landmarks, that process should, theoretically, accumulate error over time. Figure 7 tests if the authors observed this drift in the grid cell population. And again, they kind of split this into two parts, individual. Anyway, I'll get through it in a moment. So there are two pieces to this. But they looked at two groups. They looked at trials where mice found the lever quickly, so short search paths, and they looked at trials where it took longer, long search paths. They give these different colors here. So in panel A, you can see that for the same grid cells, the firing rate histograms are much sharper. That's shown here in that middle column during short searches. And the sharpness is indicated by that MVP, so the mean vector length. This during short searches, those are the blue ones, compared to long searches. So this middle column is showing firing rate histograms of lever-anchored grid cells as a function of the direction of the mouse from the lever center. So we've got these circles for the direction, and then we have lines that are indicating different rates or firing rates, so over the distribution of firing rates. Then in panel B, we see the trial matrices. So this is just by trial, all the trials in different rows. That reinforces this understanding. The further the peak firing rate deviates from the center, the greater the orientation error. And we can clearly see higher variance and lower precision when the search path is long, suggesting the internal map is degrading the longer the mouse spends in the dark. To confirm that this wasn't just an individual cell noise, so that's why I said this is really done in two parts here, because now we're going from a single cell, again, back to the population, they applied the RNN decoder from figure 5. In panel C, they compared the directional error at the beginning of the search versus the end. So beginning versus end. And you can see that the distribution of error widens significantly as the search progresses. In panel D, the probability density plot shows this shift very clearly. The distribution at the end of the search is much broader than at the start. And in panel E, when they plot the absolute decoded error against the search duration, so over time, we see a progressive time-dependent increase. You can kind of see that slope directly. This is the hallmark of a system that's accumulating error. But re-anchoring is not a reset. In 7F and G, they reach a crucial finding here. We know from figure 6 that grid cells re-anchor to the lever. So you might assume that re-anchoring resets the system, effectively wiping out previous errors. However, in these panels F and G, they show that even after the mouse reaches the lever, the directional precision remains significantly lower if the search path was long versus short. The memory of the drift is preserved. So even though the grid cells have re-anchored to the lever, they're still off by the amount of error accumulated during that search. Okay, so I'll just finish the slide and then I'll get to the question. Just one more thing is so this confirms that the grid cell orientation is updated by a path integrator that doesn't perfectly compensate for its own drift. Go ahead, Ken.
Sorry, yeah, I was going to say there's an update of the XY position in the grid cell network, but there is not an update on the head direction. This is I should frame this as a question, but if the head direction network is drifting.
Yeah.
And
that just makes sense, right? Imagine you're in a dark room and you've found a pole in the middle of the room, but you still don't know whether you rotated a bit while you were searching. Yeah.
Okay. So that's the way to. So the way to interpret these errors are kind of a drift within the head direction network.
That's what I would assume. Yeah, that was the first thought that came to mind. Yeah. Okay, so we've established that the grid cell network accumulates error over time, which we call drift. And the crucial question, we're getting towards the end here, is whether the drift is just internal noise or if it influences the animal's behavior. And figure 8 here tests if this grid orientation drift is predictive of the mouse's actual homing direction. And again, we're starting at the single cell level. The authors divided the trials based on whether the mouse ultimately homed to the left, that's orange, or to the right, green. And panel A shows the homing paths for these trials. In panel B, again, we've got this circular map giving us an interpretation of, say, they show that the lever anchored firing fields are not static. They're actually shifting their position in the firing rate map to mirror the animal's eventual left or right homing decision. To make this rigorous, the authors provide trial matrices in C. And you can see here that when the trials are sorted by the homing direction, the peak firing direction of these grid cells follows a clear trend. The panel D then is showing us the correlation between trial drift and the homing direction. And we can see a significant positive correlation. As the neural representation shifts, the animal's behavior shifts with it. Next, switching again from individual cells to the entire population using the RNN decoder. Panel E displays the homing paths color-coded by the mouse's median heading. In panel F, we see the decoded trial drift, here shown with these white circles. And you can see that the circles track with the color coding. So the color coding here, the head direction, the heading direction across the search, lever, and homing phases. The neural drift is consistently mirroring the heading of the mouse. Then in panel G, we get the quantitative support. Decoding trial drift was correlated with the homing heading error. And you can see a strong positive regression line, meaning that the error in the neural map provides a predictive signal for the actual error of the mouse's movement path. And finally, the authors wanted to ensure that this wasn't only true for select sessions. So in panel H, this shows the distribution of correlation coefficients for all sessions. And the distributions, they are all for the search path, the lever interaction, and the homing path are all significantly shifted towards positive values. And that confirms that at every stage of the task, the internal state of the grid cell network is a reliable predictor of the animal's navigational output. So kind of pulling this together across different figures. In figure 5, they showed that the map can be decoded. In figure 6, that the map anchors to landmarks to stabilize itself. In figure 7, that despite anchoring, the map still drifts over time. And here in figure 8, that the drift is functional. It determines where the mouse goes. So what does this all mean then? Traditional models of path integration have relied on the assumption that grid cells are rigid, that they maintain a stable, unchanging map of the room to provide a consistent coordinate frame. The authors here demonstrated that grid cells are not static room mappers. Instead, they're dynamic context mappers. And you can imagine that if you take this out of the mouse, for instance, this could be really important when you extend the interpretation of the effect of grid cells on hippocampal place cells that operate on contextual episodes beyond spatial mapping. This could also mean a lot, that you can have this contextual mapping and shift of reference frames, but I don't really want to go there. Just a little hint. They're flexible enough to transition from a room-based reference frame to a local landmark-based frame in a matter of seconds. And now in many analyses, the grid patterns seem to disappear in complex or dark environments, and that led to some to assume that the grid has just shut down or degraded, that it's no longer performing its function. But they showed that the absence of grid firing pattern does not mean the system has stopped working. The grid cells are still performing path integration. They've simply re-anchored their phase to a local landmark. The functions remain intact. The frame of reference has just shifted. If the grid map is shifting and translating, then how does the brain keep the math consistent? The key to this flexibility is the underlying internal geometry, the toroidal manifold, which remains invariant. Even as the grid cells re-anchor and shift phase relative to the environment, their phase relationships within the module stay preserved. This internal consistency is what allows the animal to keep performing path integration even when it switches between reference frames. Now, path integration in the dark is inherently error-prone. There's drift, and the results indicate that this drift is not just noise to be filtered out. It may be a functional signal. The accumulation of neural drift directly predicts the mouse's behavioral errors. And this suggests that the animal's navigational strategy is limited by the physical accumulation of error within the grid network, providing us with a direct readout of the animal's uncertainty. Historically, grid cell analysis required long recording epochs, minutes to hours to build stable maps. So I would say that probably their most practical contribution is the computational framework itself. By decoding grid cell activity in toroidal space, they removed the need for long behavioral epochs. We can now go ahead and characterize grid cell activity with sub-second resolution. And that opens up almost any standard memory or navigation paradigm to grid level analysis, not just for foraging tasks, but for shorter other tasks with short intervals. All of that together, it moves grid cell research out of a static map era into a dynamic real-time computational framework. And the authors showed that grid cells are not passive recorders of space, but active, flexible integrators that dynamically adjust their reference frames to landmarks at the cost of occasional drift, with the benefit of remarkable behavioral versatility. And I will get to the question right now, but I just want to point out that I also added a slide trying to think about what would a winning memory decoding study look like. So I hope we can talk about that in a moment. Go ahead, Ariel.
Yeah, just to check on the slide before, when you say it enables like sub-second grid cell readout, that's after you've done some baseline recordings, right? Still though, and you've established the grid pattern for a
So after you do the baseline recording in foraging, sort of random foraging behavior. Yeah.
Okay, cool. Just checking. I had understood that.
But okay, but it's a good point that you mentioned. Going back to this slide, maybe we should stick to this slide first before we talk about memory decoding. Ken.
And just following up on that question, the when they say and they showed earlier in the paper, they showed these really crappy during the AutoPy test, really crappy quote unquote grid stuff that didn't look like a grid at all. Receptive fields. The idea is that if they knew when, this is a question, if they knew when the when the shift had taken a place, and the amount of time that the shift was in effect for, then they should be able to get a grid pattern. They just didn't have enough time and knowledge of when it had actually made that shift to get those new grid patterns. Is that correct?
Yeah, that's my assumption. You should get a hexagonal grid pattern again if you know which reference to take, and so you'd have to know when exactly the switch is happening to maintain a nice hexagonal pattern. Yeah. Okay.
And the decoding network gets around that by assuming that, hey, this is a continuous attractor network that hasn't changed. It's just moved in X. Right? Okay.
Yeah. So the decoder is really mapping it out into something that is stable and where the relationships between the different grid cells is also stable. And so you can use that continuously. But yeah, it's a good question how to determine exactly when the reference frame shift happens.
One more question. Sorry. So this implies that there should be a reset position in the torus.
Yeah, that's what you would think. Yeah.
Is there?
Didn't see that in the paper.
Okay.
Well, then let's see. We're almost at the top of the hour, and maybe we have enough time to just think about this from the point of view of memory decoding. Just to get started on this slide, I just slapped on the most obvious, straightforward thinking that we've applied across different models, different animal models. And I thought, okay, well, since the point of winning the memory decoding prize is to be able to decode some non-trivial amount of memory from static data, you'd like to have some static data. And you'd want to be able to infer grid cell receptive fields. So the properties of what these grid cells respond to, what hexagonal pattern they're going to produce from their synapses. I took a quick peek to see how big medial entorhinal cortex actually is in the mouse. It seems to be 0.3 to 0.4 cubic millimeters. So it looks like that might be doable with today's pipelines. You'd also want to have large-scale functional recording, but maybe you don't need it. Maybe you can simplify the problem by sticking with EFIS, using these probes and focusing just on a few grid cells. I mean, the grid cells they had in this study, that was already a nice set. Because I don't know if something like calcium imaging could work here because we're typically dealing with some fairly high temporal precision. But maybe you guys know if there are other techniques that would help out here. And then, of course, what you'd love to see is you'd like to see a comparison for the application of the decoder to either the inferred grids or recorded grid maps. But there's even a bigger question here, which is, if you've got a few recordings from some grid cells that you've done with probes, and then you've got a large volume snapshot of the entire MEC, I wonder, I just don't know how in practical sense with tracing and everything, how well you can map the two back to each other. But maybe you know this, Ken. Does that seem doable?
I mean, in my simplistic mind, it does, in the sense that there should be a continuous attractor network. There's only a few ways that people have proposed to build continuous attractor networks. I was reading through one of the papers on MEC. Apparently, people believe that it might be involving the inhibitory neurons. But in any case, the idea would be neighboring grid cells that are neighboring on the torus should be exciting each other, essentially. And so the torus should fall out of the connectivity matrix, whether you have calcium data or not, and then the calcium data should match up with it.
Okay. And you feel confident that if you're tracing these connections, you're always talking about something like local proximity, so you're not ending up with, say, fibers that exit the volume and come back in elsewhere and you don't know where they are, that sort of thing?
I have no idea about that.
No idea. Okay.
I'm admittedly still quite confused. I guess I would find conceptualizing how this would work simpler if, there was a single sort of stable grid-like system that remapped when you entered new environments, that had some clear connection to head direction cells within particular environments. I'm confused as to how you can have the same system remapping within, I suppose, maybe I've answered my own questions. I'm saying this. Actually, I withdraw my point. I need to think
I
more
think
about.
the way that I think about this is like an odometer, a trip odometer on your car, and you
Yeah.
can press the trip odometer and zero it out anytime you want. And this is a two-dimensional trip odometer.
Yeah. Yeah. No, it's cool. I guess I'm not quite at the level yet, even having gone through this paper in detail, of like thinking about how to really pull out, from what I would expect to see based on if we had the connectome in front of us, how we'd go about finding, okay, like in this environment, I'd expect to see these cells connected to the grid network in this way, but I know if I was in this other environment where something had been remapped or, the animal found the lever, it would look like this other way.
The connectivity should not be changing.
Yeah.
It should be
Yeah.
a popping of back to 0, 0.
Yeah, I think a part that's still missing for me, which is maybe not important for this memory study, but a part that's missing for me is a better understanding of how the self-motion cues are actually encoded and how those appear that are related to, say, like how you're relating these to the original environment and then relating them to this new object that you've discovered, like the lever, so that you can use that and carry out the mapping. So it's part of how this input is processed into the grid cells that I still don't understand. But it
you
might not matter if we just want to have a memory prize that focuses on the decoding side of this grid cell thing.
Yeah, I mean, I feel like decoding and maybe doing a lot more work here. If the toroidal continuous tractor network fell out of the connectome, that to me that would be that would be better than the original Nobel Prize that was given for finding the grid cells. But that's just my bias. But in terms of so you've got this attractor network that has a bump of activity,
Mhm.
and you have to have a set of cells that will let's say decrease inhibition in one direction of the torus when the mouse is pointed in a particular direction and is moving
Yes.
its
Yeah, because you'd want to transition that bump over to the next set of cells encoding there, right? So that has to happen.
exactly.
Yeah.
And that also is in the connectome. It has to be.
Hmm.
Right. And so it's not just the torus that should fall out of this. It should be the torus and the things that move the bump around relative to the to those cues, and a reset would be really cool. I mean, it's all too simple, probably. And, the brain is probably doing it way more complex in a way more complex way. But the other thing is that the
So,
fly.
I'm writing this down just because I want us to not forget what would be the interesting outcomes, like the what sort of results, what kinds of findings from the static data would count as, say, oh, this definitely wins the prize. And I think this discovery of how you're shifting that bump attractor, that to me seems like a very clear and really interesting discovery, something that would, probably be prizeworthy.
Yeah.
I think much of this work would be cool. And, I think whether or not it's a memory decoding prize, it's definitely a decoding prize to pull any of this sort of stuff out from a connectome. But I have one question around memory that you might know the answer to, Randall, which is, do we know how the entorhinal cortex develops these grid connections or supposed grid connections in the first place, I asked that, like, let's say you rear an animal completely in the dark, so it like doesn't really use its visual system, or you have some other way of seeing like across development. Like, do we know if these grids come like they're genetically wired in, or they require some learning, or do we have any sense of?
I'm going to
I'm
push
trying to
this
think of
over to Ken or someone else, because unfortunately I don't know the literature on how these form during development.
I have no idea, but that is a fantastic question.
Yeah, because I'm just thinking of analogy to the visual cortex, where like you develop these orientation preferences that can be messed with if you mess with an animal's developmental periods. Is there an equivalent for the grid network?
There might well be papers about this. I mean, I can imagine that someone studying grid cells would have studied grid cells.
Yeah,
across
I was just hoping someone would know. That's it.
Yeah. Okay, we're already over the hour, but I don't want to shove everybody away, and if there are still questions, so please, if there's something you'd like to ask or something you want to contribute, we can still keep going for a bit. Ken.
I'm wondering if we might have a follow-up on a more circuit-oriented grid cell, and the developmental questions could follow from that as well.
Okay, circuits and development. I find that interesting. If we could also figure something out about the input to grid cells a bit more, maybe that's also in the circuit part, then I'd find that really interesting too. Yeah.
I've heard that the head direction network is more in the brainstem. I should look this up, but there's a reason why a lot of studies are not going for the head direction network in the mouse.
Okay. Yeah. Okay. So I'm happy to handle doing another paper on grid cells and focus more on circuit. I didn't know enough about grid cells going into this. Unfortunately, I wish I'd known more. I always thought they were fascinating, but I think now, yeah, I'm pretty comfortable with it.
Well, so Randal, you were debating, correct me if I'm wrong, fly central complex versus grid cells. Is that?
Oh yeah, I was initially thinking either I do, fly central complex or grid cells, but now that I'm on grid cells, it might make sense to continue with grid cells a little bit before switching over to something else.
I think it might, but I think it might be the case that as continuous attractors go, the fly head direction cells may be the best well-characterized circuit. And so, there might
open to being convinced to go that way.
There's an argument to be made that if you want to understand how grid cells work, look to the fly.
Learn about mouse grid cells by studying the fly. Okay.
Yeah.
Yeah, please go ahead.
I have a question about this toroidal mapping. I hadn't heard of this before. I'm also not an expert on grid cells, but has this been known for a while? And also, have people looked at, like, does it make sense to look at some of these dynamics on the torus itself?
It's a good question. I think depends on what we mean by while, first of all. It's certainly been known for a number of years that you can map to a torus. Yeah, I don't know. Ken, do you know more about this than I do?
Well, one thing that's important to understand that at least from my knowledge, there is no physical torus.
Okay, so, yeah.
Even that is kind of like, I wonder if we really know if there's any physical analog in there. That would kind of explain the developmental aspects of this. And in the fly head direction, there literally is a circle of neurons. so it's not impossible that there would be.
Yeah, so you mean for the establishing of those attractors that you would have to have input that would provide something like that toroidal space, but then mapped to grid cell input, grid cell receptive fields to end up with these attractors. Is that what
And
you mean?
you were saying about the dynamics of it, right?
Yeah.
And so I don't know, but I know that in Fly they have used optogenetics to push the attractor around. And so you could imagine, I don't know if anybody has done this, but you could imagine experiments where they try to push the position of the bump attractor around. But I don't know the literature well enough to know if they've done that.
So Ken, what you're talking about, this would the optogenetic
Yeah.
injection would, in a sense, be simulating what you would get normally from motion in the environment, where you would have the interneuronal activity be pushing the bump around. But in this case, it's because you're suppressing.
Or
I
you
suppose
could
it would be a suppression. I don't know if it would be activation or suppression.
let's say hypothetically used immediate. I don't even know if this would work, but let's say you used immediate early genes like they do in the memory tagging experiments to tag a particular position on the torus. Then you could optogenetically reset to that position, which might be an interesting set of experiments. But that would be one module, or maybe that would be across modules. I don't know.
Interesting.
It's I mean it's a fantastic system. There was a reason why they gave it the Nobel Prize. Okay, it's a fantastic system that is deep inside the brain, that is somewhat self-contained. You know, and that's what's really cool about it, because you can decode without going all the way out to the periphery, because this is supposed to be a little toroidal map, and that's exactly the type of system that we could understand with the connectome that is not going all the way out to the every other aspect of the brain.
I think I really want to now read about this, like how that mapping is done, because I'm a bit unsure if the things that I'm thinking are actually how like the map is made. But I can look up that. This.
Thank you.
Cool.
Anyone else? Any final thoughts? If not, then I want to thank everyone who showed up for being here and going through this exciting paper. And we will have another Memory Decoding Journal Club in 2 weeks. And I guess we'll find out if it's about grid cells or fruit flies or whatever else, depending on whether I'm the next presenter or maybe someone else is. Yeah, thank you all. See you later.
Thanks very much, Randall.
Thank you, Randall.
Bye.
Great.
Thank you.